Protein synthesis. It was mentioned earlier that DNA provides a blueprint for the cell structure and physiology. This refers to the fact that DNA contains the information necessary for the cell to build one very important type of molecule, the protein. Most structural components of the cell are made up, at least in part, by proteins, and virtually all the functions that a cell carries out are completed with the help of proteins. One of the most important classes of proteins is enzymes, which help speed up necessary biochemical reactions that take place inside the cell. Some of these critical biochemical reactions include building larger molecules from smaller components, such as occurs during DNA replication or synthesis of microtubules, and breaking down larger molecules into smaller components, such as when harvesting chemical energy from nutrient molecules. Whatever the cellular process may be, it is almost sure to involve proteins. Just as the cell's genome describes its full complement of DNA, a cell's proteome is its full complement of proteins. Protein synthesis begins with genes. A gene is a functional segment of DNA that provides the genetic information necessary to build a protein. Each particular gene provides the code necessary to construct a particular protein. Gene expression, which transforms the information coded in a gene to a final gene product, ultimately dictates the structure and function of a cell by determining which proteins are made. The interpretation of genes works in the following way. Recall that proteins are polymers, or chains, of many amino acid building blocks. The sequence of bases in a gene, that is, its sequence of A, T, C, G nucleotides, translates to an amino acid sequence. A triplet is a section of 3 DNA bases in a row that codes for a specific amino acid. Similar to the way in which the 3-letter code, DOG, signals the image of a dog, the 3-letter DNA base code signals the use of a particular amino acid. For example, the DNA triplet CAC, cytosine, adenine, and cytosine, specifies the amino acid valine. Therefore, a gene, which is composed of multiple triplets in a unique sequence, provides the code to build an entire protein, with multiple amino acids in the proper sequence. Figure 1. The mechanism by which cells turn the DNA code into a protein product is a two-step process, with an RNA molecule as the intermediate. From DNA to RNA, transcription. DNA is housed within the nucleus, and protein synthesis takes place in the cytoplasm. Thus there must be some sort of intermediate messenger that leaves the nucleus and manages protein synthesis. This intermediate messenger is messenger RNA, mRNA. Single quote. A single-stranded nucleic acid that carries a copy of the genetic code for a single gene out of the nucleus and into the cytoplasm, where it is used to produce proteins. There are several different types of RNA, each having different functions in the cell. The structure of RNA is similar to DNA with a few small exceptions. For one thing, unlike DNA, most types of RNA, including mRNA, are single-stranded and contain no complementary strand. Second, the ribose sugar in RNA contains an additional oxygen atom compared with DNA. Finally, instead of the base thymine, RNA contains the base uracil. This means that adenine will always pair up with uracil during the protein synthesis process. Gene expression begins with the process called transcription, which is the synthesis of a strand of mRNA that is complementary to the gene of interest. This process is called transcription because the mRNA is like a transcript, or copy, of the gene's DNA code. Transcription begins in a fashion somewhat like DNA replication, in that a region of DNA unwinds and the two strands separate. However, only that small portion of the DNA will be split apart. The triplets within the gene on this section of the DNA molecule are used as the template to transcribe the complementary strand of RNA. Figure 2. A codon is a 3-base sequence of mRNA, so called because they directly encode amino acids. Like DNA replication, there are 3 stages to transcription: initiation, elongation, and termination. Stage 1. Initiation. A region at the beginning of the gene called a promoter, a particular sequence of nucleotides, triggers the start of transcription. Stage 2. Elongation. Transcription starts when RNA polymerase unwinds the DNA segment. One strand, referred to as the coding strand, becomes the template with the genes to be coded. The polymerase then aligns the correct nucleic acid, A, C, G, or U, with its complementary base on the coding strand of DNA. RNA polymerase is an enzyme that adds new nucleotides to a growing strand of RNA. This process builds a strand of mRNA. Stage 3. Termination. When the polymerase has reached the end of the gene, one of 3 specific triplets, UAA, UAG, or UGA, codes a stop signal, which triggers the enzymes to terminate transcription and release the mRNA transcript. Before the mRNA molecule leaves the nucleus and proceeds to protein synthesis, it is modified in a number of ways. For this reason, it is often called a pre-mRNA at this stage. For example, your DNA, and thus complementary mRNA, contains long regions called non-coding regions that do not code for amino acids. Their function is still a mystery, but the process called splicing removes these non-coding regions from the pre-mRNA transcript. Figure 3. A spliceosome, a structure composed of various proteins and other molecules, attaches to the mRNA and splices, or cuts out the non-coding regions. The removed segment of the transcript is called an intron. The remaining exons are pasted together. An exon is a segment of RNA that remains after splicing. Interestingly, some introns that are removed from mRNA are not always non-coding. When different coding regions of mRNA are spliced out, different variations of the protein will eventually result, with differences in structure and function. This process results in a much larger variety of possible proteins and protein functions. When the mRNA transcript is ready, it travels out of the nucleus and into the cytoplasm. From RNA to protein, translation. Like translating a book from one language into another, the codons on a strand of mRNA must be translated into the amino acid alphabet of proteins. Translation is the process of synthesizing a chain of amino acids called a polypeptide. Translation requires 2 major aids. First, a translator, the molecule that will conduct the translation, and second, a substrate on which the mRNA strand is translated into a new protein. Like the translator's desk, both of these requirements are fulfilled by other types of RNA. The substrate on which translation takes place is the ribosome. Remember that many of a cell's ribosomes are found associated with the rough and carry out the synthesis of proteins destined for the Golgi apparatus. Ribosomal RNA, rRNA, is a type of RNA that, together with proteins, composes the structure of the ribosome. Ribosomes exist in the cytoplasm as 2 distinct components, a small and a large subunit. When an mRNA molecule is ready to be translated, the 2 subunits come together and attach to the mRNA. The ribosome provides a substrate for translation, bringing together and aligning the mRNA molecule with the molecular translators that must decipher its code. The other major requirement for protein synthesis is the translator molecules that physically read the mRNA codons. Transfer RNA, tRNA, is a type of RNA that ferries the appropriate corresponding amino acids to the ribosome and attaches each new amino acid to the last, building the polypeptide chain one by one. Thus, tRNA transfers specific amino acids from the cytoplasm to a growing polypeptide. The tRNA molecules must be able to recognize the codons on mRNA and match them with the correct amino acid. The tRNA is modified for this function. On one end of its structure is a binding site for a specific amino acid. On the other end is a base sequence that matches the codon specifying its particular amino acid. This sequence of 3 bases on the tRNA molecule is called an anticodon. For example, a tRNA responsible for shuttling the amino acid glycine contains a binding site for glycine on one end. On the other end it contains an anticodon that complements the glycine codon. GGA is a codon for glycine. And so the tRNA's anticodon would read CCU, equipped with its particular cargo and matching anticodon. A tRNA molecule can read its recognized mRNA codon and bring the corresponding amino acid to the growing chain. Figure 4. Much like the processes of DNA replication and transcription, translation consists of 3 main stages: initiation, elongation, and termination. Initiation takes place with the binding of a ribosome to an mRNA transcript. The elongation stage involves the recognition of a tRNA anticodon with the next mRNA codon in the sequence. Once the anticodon and codon sequences are bound, remember, they are complementary base pairs. The tRNA presents its amino acid cargo and the growing polypeptide strand is attached to this next amino acid. This attachment takes place with the assistance of various enzymes and requires energy. The tRNA molecule then releases the mRNA strand. The mRNA strand shifts one codon over in the ribosome, and the next appropriate tRNA arrives with its matching anticodon. This process continues until the final codon on the mRNA is reached, which provides a stop message that signals termination of translation and triggers the release of the complete, newly synthesized protein. Thus, a gene within the DNA molecule is transcribed into mRNA, which is then translated into a protein product. Figure 5. Commonly, an mRNA transcription will be translated simultaneously by several adjacent ribosomes. This increases the efficiency of protein synthesis. A single ribosome might translate an mRNA molecule in approximately 1 minute. So multiple ribosomes aboard a single transcript could produce multiple times the number of the same protein in the same minute. A polyribosome is a string of ribosomes translating a single mRNA strand. Chapter Review. DNA stores the information necessary for instructing the cell to perform all of its functions. Cells use the genetic code stored within DNA to build proteins, which ultimately determine the structure and function of the cell. This genetic code lies in the particular sequence of nucleotides that make up each gene along the DNA molecule. To read this code, the cell must perform 2 sequential steps. In the first step, transcription, the DNA code is converted into a RNA code. A molecule of messenger RNA that is complementary to a specific gene is synthesized in a process similar to DNA replication. The molecule of mRNA provides the code to synthesize a protein. In the process of translation, the mRNA attaches to a ribosome. Next, tRNA molecules shuttle the appropriate amino acids to the ribosome, one by one, coded by sequential triplet codons on the mRNA, until the protein is fully synthesized. When completed, the mRNA detaches from the ribosome, and the protein is released. Typically, Multiple ribosomes attach to a single mRNA molecule at once such that multiple proteins can be manufactured from the mRNA concurrently. Make sure to smash the like and subscribe buttons to receive notifications all new contents.